dealt with what ma? With one gene inheritance, monohybrid cross, followed by incomplete dominance, pleiotropy, multipluralism. We are left with codominance, right? And we are yet to deal with inheritance of two genes, followed by polygenic inheritance, chromosomal theory of inheritance. We will do it today. And experimental proof of chromosomal theory of inheritance also we will do it today. In our next session, we will be dealing with sex determination, genetic disorders, chromosomal disorders, we will do it. Fine, right? So, now if we see codominance. In codominance, the best example we can take is ABO blood grouping. Firstly, you have to be clear on the note of what is codominance. A gene is represented by two alleles. A gene consists of two alleles, right? If two alleles are equally expressing its phenotype, okay? If two alleles are equally expressing equally expressing their expression of phenotype, then you will consider it as what? Codominance. Best example for codominance is ABO blood grouping. ABO blood grouping, this is one expression or one character you can consider. This is expressed by one gene. What is that gene? I gene. I gene. This I gene is having three alleles, so you will consider this as an example also for, very good, multipluralism. So, this I gene has how many alleles beta? 3. What are they? IA, IB and IO, or you can simply write I. These are 3 genes of I gene, 3 alleles of I gene, okay. In how many combinations they can occur, and what is the sugar that is present in IA gene, IB gene and IO or I. So, here if you see over RBC, okay, over RBC membrane there is an antigen called as H antigen. What is the antigen that is present over the membrane? H antigen. IA, this allele will encode for the enzyme N-acetyl galactosyl transferase. This is the enzyme, okay. IA will code for what beta? N-acetyl galactosyl transferase. This will give the sugar, that is N-acetyl galactosamine, which is given to what? H antigen. H antigen will bind to what sugar? N-acetyl galactosamine. Fine, right? N-acetyl galactosamine. So, that is present for what blood group beta? IA blood group. IB, if you see, IB allele will be encoding for the enzyme galactosyl transferase. This galactosyl transferase will be encoded for the sugar galactose. This will be giving out what? Galactose sugar. Fine, right? So, IA gene will be having enzyme what? N-acetyl galactosyl transferase. This will be encoded for what sugar? N-acetyl galactosamine. IB, if you see, galactosyl transferase, this will be giving out galactose sugar. And IO, it will not have any sugar to encode for the antigen, okay. So, till here it is clear, right? So, if you consider type A, it is having A antigen, that is having what sugar? N-acetyl galactosamine. So, these plasma antibodies will be what? Anti-B antibodies. Same the case for type B blood group. What will be plasma antibodies here? Anti-A antibodies. If you consider AB blood group beta, AB blood group, they will not have any antibodies, because 2 A and B, both the alleles will show its phenotype. So over this RBC membrane you can see both the sugars A and B. Here you see O type of blood group, they will not have any antigen. Okay. So anti-A and anti-B both are present, both antibodies are present for O type of blood group. You are clear on this note, right? Now we will see maximum phenotypes and genotypes that are exhibited by this character ABO blood grouping. You are clear with the gene, right? You are clear with the types of alleles and the sugars. Did you take down? Quick. Make it fast, ma. If you take your notes properly, this will stand as a reference when you are going through NCERT reading. If you just listen and leave, na, then it will be helpless. Done? So now we will see all the possible genotypes and phenotypes. First take down all the allelic combinations. All the allelic combinations, if you see, IA, IO, IA, okay. So these are all the allelic combinations. You can see 6 genotypes on total. How many genotypes, beta? 6 genotypes. If you see number of phenotypes, here IA, IO will give what blood group, beta? A blood group. Fine. Next IA, IA, again A blood group. Next IA, IB, AB blood group. Very good. Next IB, 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 IO, both gives what? B blood group. Next IO, IO, yeah, O blood group. Fine, right? So we have 4 phenotypes here, A followed by AB followed by B followed by O blood group. As such, total 4 phenotypes and 6 genotypes. Clear, Nama? So amongst these 4, which is exhibiting the feature of codominance? Codominance, where both the alleles are expressing. Very good. Because here both the alleles are there, IA and IB, both are expressing its phenotype, that is AB blood group, which stands as an example of codominance. Clear till here? Any doubt anywhere? Yeah. Next. Now Mendelian dihybrid cross, where we learn about 2 gene pattern of inheritance. Inheritance of 2 genes. Fine, right? Inheritance of 2 genes. So in the parental generation, pure lines are taken. One is pure line dominant, that is round and yellow. Other is pure line recessive, that is wrinkled and green. Okay. So this dominant phenotype, that is pure line dominant and recessive, you have observed, right? Gametes, if you take capital R and Y, okay, and here small r and small y. Okay, these are the 2 types of gametes from parental generation. If you see in the F1 generation, all heterozygous condition you will observe, and the phenotype, if you see, dominant phenotype with heterozygous condition in the genotype. Selfing of F1 parents. Now F1 generation we will do selfing, right, according to the Mendel's experiment. So how you can identify what are different types of gametes from this? Types of gametes. How many types of gametes are formed from this? Of course you got 4, but how you got to know about this? The answer, the formula is gamete square, sorry, the formula is 2 to the power of n, where n is number of heterozygous condition. Okay, number of heterozygotes here are 2, right? So here 2 to the power of 2 will give you 4. So on total how many gametes are there? 4 gametes are there. Now you see all the different types of gametes. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. 4 types of gametes. Clear, right? And then if you observe how many zygotes are formed after crossing between these many types of gametes. How many zygotes formed? Like number of zygotes formula is gamete square beta. Okay, number of zygotes what is the formula? Take down parallelly, gamete square. How many gametes are there? 4 gametes. 4 into 4 is 16. 16 zygotes are formed. Clear? Can you see from this checkerboard, total 16 zygotes will be formed. If you are clear with this, just take a note of it. I will tell you all the combinations of phenotype, genotype and in numerical application also. Take down this. Till here, so it is clear now, beta? Yeah. Done. You have become super fast. Now you see. Tricks to remember all the possible combinations of dihybrid cross. If you observe from checkerboard, you can clearly see 9 will be of dominant phenotype, that is round and yellow. Okay, and 3 will be of round and green, 3 will be of wrinkled and yellow, and 1 will be both homozygous recessive, that is wrinkled and green. This is the phenotypic combination. And genotype, if you take that, you will see 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 1 is to 1. This will be the genotypic ratio. Okay. How you can identify the genotypic ratio of a dihybrid cross is just square on the genotypic ratio of the monohybrid cross. Okay. What you will get? 1 is to 2 is to 1 is to 4 is to 2 is to 1 is to, you will get this. Fine. Now you see all the possible combinations. So you have 9 here, 9 round and yellow phenotype of this. How many are homozygous dominant? So if you can observe from the checkerboard clearly, 1, 2, 3, 4. 4 will be heterozygous dominant for sure, and 1 is homozygous dominant, 1 is homozygous recessive, and then you see 1 is heterozygous with 1 allele. Such types you will have 2 again. So total 9 on total, you will get what? 1 is definitely of homozygous dominant and homozygous recessive. one is homozygous recessive. Here you see next 4 will be heterozygous for both the genes or both the gametes. Next 2 will be heterozygous for one and homozygous for the other. Next 2 also same condition, homozygous for first and heterozygous for second. Here it is clear, right? Next you see 3 combinations. One will be homozygous dominant and homozygous recessive like this. Rest 2 will be heterozygous dominant and homozygous recessive. Recessive though definitely it should be in the homozygous condition only. Here we go with this. Okay. One will be homozygous recessive and homozygous dominant. 2 will be of course recessive condition is homozygous recessive only. Dominant is heterozygous. Clear? Any doubt anywhere? Take down all the possible combinations, you will get numerical questions from this. At the end of the chapter, we will solve all the numerical applications. If you are clear on this number, you can easily solve. You listen it clearly first. If you are listening from between, in between if you are starting listening the genetics, you will not understand anything for sure. Listen it from the beginning and then you say. In midst if you come and say we are not understanding, how can I help you, beta? This is clear to you, no? Yeah. Take down all the possible combinations. Be clear on this note. Here 4 will be both the genes heterozygous, one gene heterozygous, rest homozygous will be 4, first and second gene like that. From this inheritance of 2 gene, that is dihybrid cross, after knowing how to identify types of gametes and how to identify the number of zygotes formed, what are all the possible combinations from the phenotypic ratio? From dihybrid cross, Mendel has discovered law of independent assortment. Okay. The third law is law of independent assortment. So, here if you see law of independent assortment, okay, it is very clear that it says like when 2 pairs of alleles or 2 pairs of genes or chromosomes, when they are segregating or passing from one generation to other generation, one pair is independent to the other pair. Okay, one pair of genes is independent in its movement with other pair at the time of gamete formation. You can simply say the segregation of one pair is independent to that of other pair. Okay? That is law of independent assortment. This happens during the time of gamete formation. What is law of segregation? That two alleles in a gene will occur without blending, okay, and they will separate from each other. There is segregation of two alleles in a gene. There is no blending in between two alleles. That is law of segregation. It is universal, and that is derived from Mendel's monohybrid cross. Law of independent assortment is derived from Mendel's dihybrid cross. Till here it is clear, right? Now you see polygenic inheritance. Okay, traits are controlled by, usually according to Mendel, every trait or character is controlled by one gene, right? But here according to polygenic inheritance, traits or characters are controlled by two or more genes, and the genes are addressed as polygenes. Genes are addressed as polygenes, and the traits are polygenic traits. Inheritance of the polygenic traits is known as what? Polygenic inheritance. Okay, and this is also known as quantitative inheritance, because pair of dominant recessive alleles in total, many genes are coding for one character. So quantity of genes is more, as such we will call it as what? Quantitative inheritance also. Polygenic inheritance also addressed as quantitative inheritance. Genes involved are polygenes. In these genes, whichever dominant alleles are there, okay, dominant alleles both in the homozygous condition or in the heterozygous condition, they will give combined effect. That combined effect of gene expression is known as additive effect or cumulative effect. For example, you see skin color in humans, height in humans, kernel color in wheat, and pollen tube length also. Okay, this will be variable. There are not just two traits that are expressed. Okay, there are not just two variations that are expressed, many variations are seen. Okay, so clear, right? Traits are known as polygenic traits. Inheritance of polygenic traits is polygenic inheritance, which is also known as quantitative inheritance. Genes are polygenes. This will show additive or cumulative effect. Examples include skin color, height and kernel color in wheat. Take down, quick. Already there, very good. Next you see, so these are three laws of Mendel's inheritance. What are the deviations if you observe? Deviations of Mendel's law of dominance. Incomplete dominance, codominance, multipluralism and pleiotropy. From law of independent assortment, deviation is polygenic inheritance. So this will be phenotypic and genotypic ratio. I hope this table is not there with you. Take down this table. I know table won't be there because I designed it. Monohybrid cross, phenotypic ratio is 3:1, genotypic ratio is this. Dihybrid, phenotypic, genotypic ratio. Test cross, incomplete dominance, genotypic, phenotypic ratio will be same. Codominance also, it will be the same. Polygenic inheritance, the ratio is this, which will give you bell shaped curve in a graph. When you plot that gene expression in a graph, it will be bell shaped curve. Take down. If you are done with this, then we will begin with chromosomal theory of inheritance. Done ma? Now you see, most important formula that we are going to apply. Of course, I have told you two applications. One is to identify types of gametes, like how many types of gametes are involved in formation of zygotes. Okay, that is what 2 to the power of n, right now beta, where n is equal to number of heterozygotes that are formed. Next is number of zygotes that are formed will be what? Gamete square. Number of phenotypes and genotypes. How to identify how many number of genotypes and phenotypes are formed? 2 to the power of n and 3 to the power of n are the formulae. But this number of phenotypes and genotypes formulas will vary for multipluralism and polygenic inheritance, where n plus 1 is for multipluralism, n by 2 into n plus 1 for genotypes, to identify number of genotypes here. Here it is 3 to the power of n and here it is 2n plus 1. Always n will be equal to number of heterozygotes condition. Okay, take down. This formula chart is very important. Fine, right? Clear till here? Yeah, next. Here we go with chromosomal theory of inheritance. Before going with chromosomal theory of inheritance, we have to discuss about few points, that the Mendelian law of inheritance, that is law of dominance, segregation and law of independent assortment. Though he has finished his experiments in the year 1856, he has published, the publications of Mendelian's principles are in 1865. Fine, right? He has published in limited publications, that is in Bryn Society of London Limited, some Bryn Society of London. In that paper he has published his experiments. Okay, 1856 he has completed, 1865 he has published, but then it took so many years for recognition. The work got recognized, in the year 1900. Work recognized in the year 1900. Okay. So, how it got recognized after so long? What are the main reasons? First thing is, there are limited sources of publications. Okay, limited sources of publications where he has published in only one journal. Second thing is, his mathematical and statistical approach was unconvincible by the competitors there. Okay, contemporaries there, they did not approve the mathematical and statistical approach. Second thing, his point that factors do not blend. Okay, they are stable. Factors according to him is what? Genes. Genes are stable, he is saying, but at that time Charles Darwin is driving with his concept of evolution, okay, evolution and variation, and here Mendel is saying that factors occur in pairs and they are stable. Okay, this is unconvincible again at that time. His mathematical approach failed it, and there is no clear evidence to show the segregation, chromosomal segregation and all. In 1884, with the development of electron microscope and all, 3 scientists were rediscovered the Mendel's law of inheritance. Who are they? Von Schermach, De Vries and Carl Correns. Fine, Von Schermach, De Vries, Carl Correns, they have rediscovered Mendel's pattern of inheritance, and they have studied cell division in detail. Cell division in detail. So here if you see the chromosomal study, what we have learnt in cell cycle, G1 phase, S phase, DNA duplication will occur, and this will be G2 phase. Okay, and in meiosis 1, especially in the zygote stage of prophase 1, you can clearly see the bivalence or chromosomal diploid chromosomes, that is bivalence, you can see synaptonemal complex formation. In zygote stage of meiosis 1, sister chromatids will segregate apart during anaphase, and in anaphase 2 chromosomal splitting takes place. That is how you can see 4 germ cells are formed which are haploid germ cells which are formed. Clear, right? G1, G2, meiosis 1 zygote, meiosis 1 anaphase, meiosis 2 anaphase and germ cells. So, this is the rediscovery of Mendel by 3 scientists. Fine. Now, they got to know what is the cell division, how is it happening, okay, what will be the chromosomal movement and gene movement. As such, they have discovered this. Okay. Now, based on Mendelian pattern of inheritance and chromosomal segregation, 2 scientists, that is Walter Sutton and Theodore Boveri, they have united the combinations of chromosomal segregation and Mendelian pattern of inheritance, and they have given us the chromosomal theory of inheritance. What did they note according to these 2 laws or rules? That behavior of chromosomes was parallel to behavior of genes. Behavior of chromosomes is parallel to the behavior of genes. The pairing and separation of one pair of chromosome would lead to segregation of a pair of factors. Fine, right? Sutton united the knowledge of chromosomal segregation with the Mendelian principles. And he called it as what? Chromosomal theory of inheritance. Here you see chromosome, this occur in pairs. Genes also occur in pairs. Here segregation occurs at the time of gamete formation, such as only one pair of gene is transmitted to the gamete. Here gene, if you see, segregation occurs at the time of gamete formation and only one of each pair is transmitted to the gamete. This independent pairs segregate independently of each other. Here one pair segregates independent of the other pair. Clear with chromosomal theory? Next. What is the experimental proof for this? Again, an experimental proof is required. The experimental proof for chromosomal theory of inheritance is given by Thomas Hunt Morgan, who is known as what? Father of experimental genetics. He didn't just give the experimental proof of chromosomal theory of inheritance, but also he has discovered the concepts like linkage, crossing over, sex linkage, crisscross pattern of inheritance. This and all he has discovered with us. So, he is also known as what? Flyman of genetics, as he has worked out all these discoveries on Drosophila melanogaster. Okay. So, why he has chosen only Drosophila? That doubt we will get, right? Why Mendel has chosen pea plant? We have got all the reasons. Now, we should be clear with this note also why Drosophila is chosen by Morgan. It is because it is easily available on the fruit surface. So, it can be easily reared, right, inside a bottles with yeast cultures. Second point, a new generation can be raised within 2 weeks, just 2 weeks, with a single mating event producing hundreds of individuals. Okay. And then females, if you see, females are easily distinguishable from males because they are having large size and ovipositor, because female body is having large size with an ovipositor. Okay. Male heterogamety is XY. Transmission of heteromorphic chromosomes can be seen. So, what type of heterogamety is expressed by Drosophila? Male heterogamety, where females will be of same chromosomes XX, male will be having XY. Clear? These are the reasons why Drosophila is chosen by Mendel. Now, here there is an experiment which is conducted to identify link. From this experiment, two discoveries were done. One is linkage and second is recombination. Based on that, Alfred Sturtevant given us chromosomal mapping. Three things were done later. Okay. First, two types of crossing overs were done. This is cross A and this is cross B. What are the traits that Mendel has considered for both female Drosophila and male Drosophila? Traits include, take down, this is not there in your NCERT. If you take down, this will be reference when you are reading NCERT. Okay. Traits include body color, eye color, wing shape. Body color, eye color and wing shape. In this you see both dominant allele and recessive allele. Dominant allele and recessive allele. So, recessive allele, Mendel has given the alphabet according to dominant allele. Here there should be some uniqueness, no. So, he has given the alphabet according to what? Recessive allele. If you see body color of Drosophila, brown is dominant phenotype, yellow is recessive phenotype. So, it is Y and Y plus, this is the denotion. Okay. Eye color, red is dominant, white is recessive. So, it is W, W plus here, this is the denotion. Wing shape, normal wing shape is normal, is dominant, miniature wing shape is recessive. So, it is M, M plus. Clear? This is all about what? Traits. So, now if you have to plot it on a chromosome, here we go. Yeah, female and male. He has given all the dominant phenotypes for female or male only. Yeah, two alleles on the same locus. This forms homologous chromosomes. Here you see Y, W, M, Y plus, W plus, M plus. Okay. Over the DNA, genes are present. What are chromosomes? Condensed form of DNA. So, here if you see beta, the distance between body color and eye color is very minimal. Okay. Less the distance, less will be crossing over. Less will be the possibility of crossing over because they are linked. Okay. Linkage is a concept where you will learn about complete linkage, partial linkage later and all. Here you remember one thing, that if two genes are located close to each other, they are linked. Okay. There will be less possibility of recombination or crossing over and more possibility of linkage. Same the case you consider with eye color and wing shape, the distance is more. More the distance between the genes, more the possibility of linkage, more the possibility of recombination, less the possibility of linkage. These three things are clear to you know. Here you observe in cross A, if you see beta, two things are considered, two traits are considered, body color and eye color. As they are closely arranged to each other, after the parental cross in F1 generation, if you see percentage of parental type combinations and recombinant combinations, parental combinations are 98.7 because they are closely linked to each other. Recombinant types will be 1.3%. Same in the case of cross B, if you see, parental combinations will be less because it is the cross in between wing shape and eye color. Okay. Recombinant types combination is 37.2, parental combinations will be 62.8. Okay, parental combinations how many? 62.8, recombinants will be 37.2. Maximum recombination percentage will be 50%. Remember that maximum recombination is how much? 50%. 50% parental combinations, anyhow it will be there. Clear with this? Take down this diagram and this two. It will be easy for you when you are referring the NCERT. Done, ma? Excuse. Now, from this concept after cross, what two things are discovered? One is linkage, second is recombination. Excuse me. So, about this chromosomal mapping, we will look into the next class, chromosomal mapping, sex determination and genetic disorders. We will finish it off and practice NEET PYQs as well. So, come prepared with these concepts. Okay? That is all for the session, ma. Thank you.